David, in the most simple terms, what is RASP?
So RASP stands for Robotic Antimicrobial Susceptibility Platform. Antimicrobial susceptibility is the process in the laboratory of determining how susceptible a bacterial isolate is to a range of drugs. It's a phenotypic test.
And what is RASP designed to do?
RASP is designed to take what used to happen in the lab under a manual process and to speed it up and to scale it up so that we can look at large numbers of isolates much more affordably than we could in the past. It also allows us to perform so-called standard surveillance. This is the surveillance that is being performed internationally under the guidelines issued by various standards authorities, so we get the best quality data to international standards. So putting it all together, large numbers of samples, large numbers of isolates, high standards of results meeting international standards, but also can be used for clinical work as well.
Why was RASP developed in the first instance?
So we developed RASP because we fundamentally believe that for practices to change in the antimicrobial stewardship, antimicrobial resistance ecosystem, the people who have the greatest power to do that are farmers and veterinarians. And they need data that is specific for their circumstance. And this wasn't happening with national surveillance programs, which not only were they very small and I believe very inaccurate or had a significant problem with accuracy, they were also very slow to be completed. So with RASP, we are able to address herd level resistance issues across quite a number of herds and to turn that data back to the people who matter, i.e. veterinarians and producers, within a reasonable time frame so they can actually take action on it.
What bacteria are used for the RASP MIC protocol?
Okay, so RASP can actually work on any culturable bacteria, okay? But we tend to focus a lot in veterinary and food applications of AMR surveillance and assessment on E. coli and a few other bugs. But it's really been worked up on commensal E. coli. Nevertheless, it could be used for pathogenic E. coli, staphylococcus, a whole range of pathogens. But the biggest focus we have is on commensal E. coli. And the way we do that is we can isolate commensal E. coli from the feces of farm animals. So pigs, for example, have anywhere between 10 to the power of 6 and 10 to the power of 8 E. coli per gram of feces. So it's a huge amount in there. And we have developed ways on robotics of getting those isolates out from each sample or from a pool of samples and spreading them out on an agar plate and picking off the number we need, and therefore getting coverage across a reasonable number of animals in the herd. And this handling of isolates is actually all done robotically. People might remember working in a microbiology lab with a platinum loop, but it's quite a tedious task. And if you think about the conventional microbiology laboratory, that process is quite onerous if you're asked to do lots and lots of isolates. By going to a robot, we can do it quickly and more efficiently and without much of the error that entails manual handling of isolates.
Why is commensal E. coli a good choice to look at when you're looking at surveillance for antimicrobial resistance?
Right, so if we look at it biologically, commensal E. coli is ubiquitous. As I've said, it's present in high numbers in virtually all animals, all terrestrial animals, and pigs are a very good example. There's a huge amount of E. coli in the gut of pigs. So it means we can test any pig at any farm at any time for a start. And we don't have great hassles about finding the bugs. When you're talking about resistance in pathogenic organisms, of course, you've got to have a disease outbreak or cases, and that can be very hard to get all the isolates. And sometimes the isolates are actually hard to procure from natural disease cases. The other issue biologically with E. coli in general is that it tends to respond very well to the selective pressures of antibiotics that enter the system in which they live. So if veterinarians and producers are using antibiotics, E. coli will respond. And therefore, it's reflecting back to us what type of drugs have been used historically in that herd. It's probably also worth noting that if we go over to the sort of more man-made side of issues, there's been a lot of international agreements about what bugs we should use in surveillance. And E. coli is the most common ingredient of all the surveillance programs around the world. So it's a very well recognized bug for basing measurements and performing comparisons with.
Mm. And how do you measure resistance in those commensal E. coli's that you acquire through the robotic system?
We have a number of ways of doing it on RASP, but the main one to talk about, I think, is based on establishing the minimum inhibitory concentration of drug. And this is the bee's knees from the point of view of assessing phenotypic resistance. It's the best assay that we have, and we're doing that on the robot for that reason. The way it works is we use 96 well plates and we grow up an isolate to a standard density, and it's inoculated into these standard 96 well plates. And in those plates are a panel of drugs. There might be, for example, 12 different drugs in that plate. Now, those drugs are present in multiple wells because we need to get a graduated scale of concentration. So we might have ampicillin present at, for example, 2, 4, 8, 16, etc. And we inoculate the drugs into that whole plate where there's ampicillin, there's tetracycline, and there's ciprofloxacin, etc. And then we assess, after that bug has been inoculated into that plate and given a chance to grow in the incubator, we assess the lowest concentration at which that bacteria will grow in the presence of each of those drugs. And therefore we get a result for each bug, we get a result for each drug that is on the plate. And that's the minimum inhibitory concentration.
And how do you choose which antimicrobials are part of the panel for the surveillance work?
So the panels are constructed. In the past, we've tended to rely on advice from a group of experts. And in some surveillance systems, these are standardized panels. The EU, for example, and the US system, they all use a standardized panel. And that's been agreed to by their experts. Typically here, we've got together with people in the industry and in government, and we've been considering things such as the drugs that are used in that particular production system, trade-related matters in terms of wanting to have data that's comparable internationally, and also the drugs that are used in human medicine, because a big driver on all this AMR work has been public health. So putting all that together, a group of people come up with a panel, and that panel includes not just the drugs, but which concentrations they are present at.
Mm. And then when you're reporting out the results that you get from RASP, how is the resistance weighted for each antimicrobial?
Okay, so one of the most important, I think, developments in recent decades on antibiotics is actually the classification system that basically says some drugs are more important than others when it comes to human health. And this was a process developed and adopted in Australia and also internationally. And so we have rating systems. And the way we use those rating systems is we can see that drugs like fluoroquinolones have a much higher importance, so that would be drugs like ciprofloxacin, than drugs like penicillin, which have a lower importance. So in order to simplify the results and to convey in a simple one number measurement to producers and veterinarians, we've developed an index, much like a share market index, where a share market index aggregates information from all the companies on the share market and the bigger companies having a bigger influence. In this case, we develop an AMR index in which we aggregate the data for all the drugs in the panel. But the drugs that are of higher importance, like fluoroquinolones and third generation cephalosporins, they have a much higher weight in that index than drugs like tetracycline and ampicillin.
And how does the degree of resistance in the isolate inform the weighting? If it's a multi-drug resistant or an extensively drug resistant isolate, does that impact the weighting?
Yes, it does. So the more resistance an isolate has, the higher its index. It's a very simple index. Basically, we look at a drug, at a bug, and we say, okay, what resistance is present? And we go, okay, ampicillin, that has a weighting of 1. Let's say carbapenems, that has a weighting of 4. And we are multiplying, adding up those indice weights to get the overall index for the isolate. And we can then take that isolate level index and we can take an average to give an idea of what's happening in the herd. But we also report for herds, we also report the spread of isolate index. Now, this isn't throwing away anything that we used to deal with. It's important to say that we still have in the background all the data that people are familiar with if they've been doing a lot of work with antibiotics and resistance. That data is all there and it's reported. We use the index because we realize that when you start reporting for a lot of bugs in a lot of herds and a lot of drugs, that it gets very complicated. And this was a great way of simplifying and allowing some much more easily digested comparisons between, for example, herds and sectors perhaps, and time periods.
What data do vets and producers get back from samples that they send in for the MIC surveillance work?
Okay, I think it's important to say that in the old way, in the old system, they get nothing. They literally get nothing, although we had been working very closely with in Australia with R&D corporations such as Australian Pork Limited. And in the early days, some of that data got back to producers, often with a couple of years delay. Okay, but now what we're sending out is a multi-page report. It's 3 or 4 pages long. And we give the overall data at the front in the very simplest form. For example, that would be the herd index value, the mean of all the isolates index on that from that herd. And then as we go down, we start to reveal more detail on the individual isolates, what forms of single resistance are present, what forms of multiple resistance are present. And then we allow, if it's within a project setting, we can show the producers how they look compared to other herds. This is done on an anonymous basis. So it allows herd-to-herd comparison, which is a type of benchmarking exercise, which provides people an understanding of where they sit in a sector.
And that data is all de-identified as well, isn't it?
Absolutely. There's no point really in revealing identities in this data. We actually don't know them in the lab because we work through an intermediary. And we put out those results because without some basis of comparison, people find it very difficult to understand where they are, what they should do.
And that's great because it's similar to what they do on the human health sphere with hospital surveillance for AMR as well. So it's really nice to have a comparative program available in animal health.
I think that a lot of producers in different parts of agriculture are used to this on production indices, on levels of protein in grain, levels of protein and fat in milk and back fat and all that sort of stuff. A lot of that data is often shared in different industries. So this is along those lines, so people know where they stand.
What is your advice on how the data that is then reported back is used by vets and producers on a farm level? And you can be generic.
Okay, so the way I think RASP output should be used. Now, the typical RASP output with the MIC test has, usually somewhere between 90 and 96 isolates. And they'll get a pattern of resistance across all those isolates. And the main thing is to understand which forms of resistance are present. in what we would call single resistance. Like, do I have resistance to penicillin drugs like ampicillin? Yes, no. Invariably you do in, for example, the pork industry. You may not in an extensive grazing segment industry, and so on. You're looking through. And particularly when it comes to drugs of high importance that we talked about before, then this is starting to suggest in a type of preventive action in terms of suggesting whether you should be relying less on particular drugs that might actually impact the herd's disease control or the marketability of products in the future, based on the presence of resistance to high importance drugs. So we would encourage people to sit down and look at those results and take it as an accumulated summary of the effect of all the use of antibiotics in the past and other factors around stewardship, like the importation of animals from other herds, the exposure to other species, including humans. We know that's actually quite important. And that's what this data is telling you. It's telling you the summary effect of all those factors. And based on that, decide on how or if you want to modify antimicrobial use in the future against specific pathogens. It might be important to say that it doesn't avoid the need to culture and sensitivity, appraise the sensitivity of pathogens. Just because you've done RASP doesn't mean that you don't have to do, if you have an E. coli scours problem, that you should also look at the sensitivity of those pathogenic isolates. But it's starting to give you intel about where you're going and the summary effect of all those selection measures. I think it's a very good way of motivating and understanding why you want to do stewardship. It doesn't have to be done religiously. We don't think it should be done every year, but certainly at regular intervals longer than a year, it could be very beneficial for telling people where their antimicrobial stewardship is going.
Yeah, that was going to be my next question, I suppose. If a producer and a veterinarian in unison make changes looking at their, all their herd health data in conjunction with that RASP report, obviously, and do make changes, how long would you anticipate those changes would need to be in place to see a meaningful change on a follow-up RASP
Well,
report?
this is a fabulous question. It's one of the it's a $64 question in antimicrobial resistance. It's a phenomenon called reversion, or in the longer phrase, reversion to susceptibility. How long does reversion to susceptibility take, and what are the conditions needed for it? We don't have many answers for that. We do know that reversion can happen. I can see data from the chicken sector in this country, based on data collected in about 2003 versus data collected in about 2016, that there was some reversion to susceptibility in some bugs. It does happen. But the jury is still out about if and how long reversion takes. But we do know the continued use of drugs where resistance occurs will elevate the level of resistance in the microbial population. But in terms of dropping it back, it's still unknown. We'd all like to think that we could stop using antibiotics and we'll go back to zero. However, if you think when we've looked at animals in remote locations grazing, we still find resistance in rangeland goats in Central Australia and in rangelands where they're not being exposed. So we do expect resistance, some forms of resistance, to hang on very tightly in microbial populations. And I think we are, with this technique, it's actually a great technique for study reversion, because it looks at large enough isolates to detect it. But we still haven't got the answer to how long it takes.
How can RASP help with early detection of emerging resistance?
Okay, so there's a second variation of RASP called RASP Quant. And what we've been doing with that is, instead of looking and working entirely with individual colonies all the way, we do what's called agar dilution. And that's where we have in the agar plates that we inoculate diluted feces onto, we have an antibiotic present. In fact, we have 5 different plates, each with a different antibiotic. And what we do is we dilute the feces to a number of dilutions and we plate it out robotically onto these plates. And the great thing about this technique is instead of looking at 90 colonies or 100 colonies with MIC, which is more expensive, we're looking at a vast number of colonies each sample. So I've said before, we've got about 10 to 6 to 10 to the 8th E. coli per gram. And if we're not inoculating, say, 0.1 of a gram on there, so we could be looking at 10 to the 5th E. coli every sample, which gives us really high level of sensitivity, far surpassing all these other tests that are in use because they evolved for clinical purposes. I have this isolate, it's a pathogen, and I want to know the answer. That's not what we're doing here. We're trying to understand, is resistance present at very low levels or very high levels? And so this is a highly sensitive technique for detecting whether resistance is present. And that's what we've demonstrated in some of our work, that it's very, very good at early detection of resistance in commensal E. coli.
And so does that protocol then provide an opportunity to give evidence of freedom from particular resistances for market access? Is that a potential use for that as well?
It is a potential use. I sense that people haven't been keen to go down the route of market access, which is a bit of a minefield in a number of ways. It's a difficult problem to deal with for industries and government. It's very complex, and we really need to be certain of what's going on. But in the future, I think there will be more emphasis on market access type issues around AMR. And in Australia, we have some advantages there because we didn't register some drugs like the fluoroquinolone class. We didn't register them for food animals. We have fairly constrained use registration requirements for third generation cephalosporins, and that includes ceftiofur. However, in order to demonstrate in a commercial sense or a government sense, if that's what we wanted to do, then this technique is ideal for doing that. Proof of freedom, a proof of absence of a drug resistance or any disease is a technically complex and difficult task, but a lot of it depends on the sensitivity assay. And I think in this assay, we have one of the best, if not the best.
What happens if an unexpected multi or extensively drug-resistant result was to come up on your RASP MIC? Is there follow-up that you can do to try and figure out where that's come from? Is it something that you just flag to note in future?
Okay, so if we were really concerned about a type of resistance coming through either the MIC pathway or the quant pathway from RASP, we could take those individual isolates and we could separate them out in a separate pathway in the laboratory and explore them. We could redo the MIC if that was necessary, if we thought that was appropriate. We could extract the DNA and go down a genomic pathway, which we are doing this now routinely in surveillance. In some cases, we're doing it to all isolates because the cost of sequencing has come down so much. Sequencing data, although much celebrated, it's still very complex data, particularly when you're looking at multiple genes. And so it's still worth doing. And it does help us understand, for sometimes, the heritage of the isolate. Does it have characteristics, for example, sequence type, that suggest it came from a particular host or place in the world, etc. So those sort of things are very possible now. And we do them routinely now for isolates of concern.
Is there a role for RASP in a clinical disease outbreak situation, and what might that look like?
In some circumstances, we get new diseases or very unexpected events. And we've seen this and we have worked on these sorts of problems where a bug has turned up. It's a bug that we know can turn up, but the AMR pattern expressed by that bug is of concern. And so in order to get our heads around some of those issues, we've gone in and we've got not just commensals, but we've actually got large numbers of pathogens, because in outbreaks where there's no control present in particular, you can actually get quite a few pathogens if you really try. And so we can go in there, collect lots of swabs and work on a bug like Staphylococcus, for example, would be a good example. And instead of characterize one or two, which would be the situation, the standard situation, and now we can look at several hundred. We can create a library of such isolates, and we can subject them to any phenotypic or molecular test we like. And typically in some of these unexpected outbreaks, we would be looking at sequence type as well as resistance patterns and trying to understand, where did this bug come from? How should we deal with it from an AMR perspective? And when you're dealing with the numbers at this level and bigger herds or flocks, what the use of RASP means is you have a great deal of confidence about your advice and the action you're going to take. And I think that's been a process well received by people who've had those sorts of outbreaks.
Some practical questions for vets. I guess, particularly again, looking at your RASP MIC, what samples are submitted, and are there any specific protocols or recommendations for on-farm sampling?
So RASP MIC, all we really need is a swab in transport media, a fecal swab in transport media. And what we've been tending to do in order to get a good representation of the E. coli in the herd is we've been taking anywhere between 10 and 30 swabs per herd. And my feeling is the more swabs you take from the target group that you're interested in, the better, up to a point of practical limitations. So when you get those swabs, we can pool them in the laboratory. At the moment, we're looking 90 to 100 isolates in that area, and that gives us a certain amount of sensitivity at a certain cost. We can increase the number of isolates and increase the cost, or reduce it, and so forth. But we've been working in that target of about 96 isolates, and that is a good way of doing it. But the swabs in transport media are great. You just go into the shed, you swab the feces, stick it in the transport media. You can get this job done very quickly, and the samples go off to the laboratory like that, because the packaging is very simple. You need to arrange with the laboratory in advance. It's very important that samples like this do not turn up on the doorstep unannounced, so you contact the laboratory. Similarly with RASP Quant, which is something that we have used less, but we also think is very powerful. If we're going to calculate the number of organisms per gram, this is what we like to do, then we need a volume of feces. And so we use yellow top type containers, and then they have to come chilled to the laboratory. Again, it's something to discuss with the laboratory before you set off in that direction.
Mm. And I think most people will be familiar, but the laboratory is located where?
So the laboratory is located at Murdoch University in Perth, and it used to run under the banner of Murdoch University as a research lab. It has now a commercial spin-off called Aquila Scientific, and the reason that was done was to avoid some of the constraints around setting up projects every time a sample came in.